The broadcast is now starting. All attendees are in listen-only mode.
Well, good afternoon, everybody. This is Matt Godson, co-CEO with Momentum Events, and it's my pleasure to welcome you here today to this afternoon's webinar entitled Working the Science and Regulations Harder to Win Your Drug and Medical Device Case. Before I introduce today's presenters, I have a little housekeeping. Firstly, this session is being recorded, and everybody listening to the presentation right now will receive a link after the session that will enable you to review and share the recording, audio, and visual of this session. In addition, the presenters have agreed to make the slides available after the session. So once again, in the follow-up email that you will receive probably within 24 hours or so, you will receive a link that will enable you to download the presentation. There will be a Q&A after today's session. Please use the questions and answers panel in the GoToWebinar panel to the right of your screen to pose your questions. With that being said, it's my pleasure to hand things over to the presenters this afternoon. We have John Clark, April Zambelli-Weiner, and I'm going to hand over to David H. Schwartz for the balance of this presentation. David, over to you.
Hello and welcome. Thanks to Matt and to Momentum Events for organizing this webinar and promoting the event. My name is David Schwartz. I'm a founding partner of Innovative Science Solutions, and we're a scientific consulting firm that specializes in working with counsel on legal cases involving complex scientific issues. So I'd like to provide a brief introduction to the webinar today and then introduce John and April. So we're all familiar with the big drug and device cases that have been waged over the years. This slide just depicts some of them. Needless to say, these are high-stakes cases. As an example, although an old example, in the Fen-Phen litigation, there were $6.44 billion class action settlement with over $500 million in plaintiff awards. This is just a brief schematic that I put together that summarizes the conceptual aspect of a drug and medical device case. Now, in order to develop the most defensible strategy, you must have a facility with the science and the regulations that guide the science. When all of these pieces are put in place, you're in a position to get great testimony. Here's a short extract from a deposition of a plaintiff epidemiologist taken by my friend and colleague Nathan Shockman in the SSRI birth defect litigation. What I love about this particular set of testimony is the last Q&A where Nathan says, and to paraphrase my cousin Vinny, no self-respecting epidemiologist would do a meta-analysis by including the same study twice, correct? Well, that was an error. Yeah, you're right. So this was a deposition of Dr. Anik Berard, and it shows how complex epidemiological data and methods can be ultimately boiled down to a simple error easily understood by a jury. So at the end of the day, most of the principles we'll discuss this afternoon involve an assessment of causation. There seem to be three broad possibilities with respect to causation, and I've sort of outlined them here. So first, a drug or device is not capable of causing a condition, and this would be a general causation defense. Next, the drug or device can cause the condition, but didn't in this particular case, and that would be a specific causation defense. And finally, the drug or device can or did cause the condition, but the warnings were adequate. So this would be sort of a product misuse kind of situation. So when confronting a new drug or device case, it's critical to step back and make sure you know which situation you're arguing to sort of make the clinical data align with the particular arguments you're making and the causation question. So what kind of data do you grapple with as a drug and device litigator? So here's a typical scenario. The product first goes through discovery. You can see down here. I think you can see my cursor. And this involves preclinical studies in animals and in vitro testing. Then comes the clinical development phase, and ultimately the FDA, the approval and launch. And in many cases, a litigation will be filed sort of in this particular area. All of the study types listed here will likely come into play in the litigation. So the gray bars represent the studies performed by the sponsor. The light blue bar represents spontaneous adverse event reporting and reports to FDA. And then the red bars represent independent studies that often form the heart of sort of the litigation battles. And these studies and sort of this whole set of studies will form the landscape that April and John will be talking to you about today. Okay, so let's get to it. So both Dr. Clark and Dr. Zambelli-Weiner are good friends and colleagues of mine. I've worked with them for many years. Dr. Clark has worked as a medical expert in healthcare product development and monitoring for 25 years with a focus on safety signaling, product complaint analysis, risk management, safety surveillance, clinical trial development, pharmacoepidemiology, and marketing outcomes. John is currently the President and Chief Medical Officer at PCS Global. Dr. April Zambelli-Weiner is a senior epidemiologist and biostatistician with over 16 years of experience in providing scientifically rigorous research and consulting services to diverse clients in both the public and the private sectors. April has consulted for the NIH, the CDC, and for many Fortune 500 companies. She's supported science and testified in drug and device cases for both plaintiffs and defendants. And April is president and founder of Translational Technologies International. So with that, let me hand it over to John, who's going to talk about making effective use of safety surveillance data in drug and device litigation.
Well, thanks to David for that introduction. Next slide. So I want to start out by talking about what we call traditional pharmacovigilance, which is case-based systems. I think we're all aware of these systems, been around a long time, since the 1960s, post-thalidomide. And these systems are really part of a worldwide network that acquires what we call spontaneous reports. They occur in 3 levels: at the company level, at the national level, such as the FAERS database here in the U.S., and at an international repository, which is put together by the WHO. It's located in Sweden. They're essentially connected together, can be zipped around the world using the ICH E2B electronic transmission standard. They come at you from all directions. Over on the right-hand side, studies, literature, etc. So they're chaotic databases, but in some respects, that's actually good. And I want to emphasize, cases do contain important causality information. It's not definitive, but they give us a lot of information that people like April can then subsequently use in studies. That central oval there is really important. The reporting pattern of multiple cases is oftentimes more important for signal evaluation and identification than anything else, such as its pattern over time or its pattern over geography. Next slide. So, important point, safety signaling is a process. It is a process that assesses multiple cases. It starts with multiple cases and then proceeds to other kinds of information. We often use the term disproportionality, which is not the same thing as causality at all. All it means is, in our case reporting systems or some other system, cases are occurring more often than we would have expected. So, for example, if we get cases of heart attacks in 25-year-old women, you don't need a lot of those to be disproportionate. If you're getting heart attacks in 65 and 70-year-old men, you're going to need a lot of cases to get disproportionate. A threshold is simply an arbitrary setting with a sensitivity, specificity setting that defines safety signal in the sense of a disproportionality. So it's the starter for the process, but the process is much more complicated than that. Going down to the 4th bullet, then, most people know about signal detection, the first phase of signaling, where we identify the signals. But many people are not aware that there's a lot that has been written and put into regulations as well, mostly in Europe, about signal evaluation. And signal evaluation is where you take that case series, start extracting the basic information, associated with that signal and then take it from there as necessary. There are essentially three different kinds of signal detection in a very broad sense. The first one is to look at cases. Now, I know people probably are thinking, gee, are we still using cases? The answer is yes, we are. And in a minute, I'll show you the particular type of case that we're looking for. Many cases don't help us at all. There are some cases that help us immensely. The type 2 that I've listed here is essentially data dredging through a case reporting database. And the third is some kind of statistical disproportion that occurs in a more formalized study. That study can be either human or animal. Important point, type 2 methods, data dredging, will get you a lot of false positives. So the real key in many safety signaling systems is to work them up sufficiently to know whether or not you're dealing with a false positive. I'll give you some examples of that. And then, of course, all of this may or may not be followed by studies. Next slide. So I think many people at this point would say, well, let me go take a look at the CFR. That will tell me all about safety signaling. And unfortunately, it won't. What you'll see in the CFR, either on the clinical or the post-marketing side, are some very general statements, such as you're supposed to assess all safety-related information, no matter where it comes from all sources, and let the agencies know when there's a problem. Well, that's not really a standard. That's more kind of a philosophy. About the only relatively specific standard is on the clinical side, and that pertains to a case series, a very particular type of case series that originates with an expedited report, and it is called a similar AE analysis. Other than that, there's really not anything in the way of signaling methods that are described in the CFR. Next slide. So where does all this stuff come from? And the answer is two organizations which are called CIOMS and ICH, and I've listed here what those names stand for. CIOMS was started back in the late 1940s. It's actually part of the WHO, which is part of the UN, and for most of its existence, it held a lot of conferences that didn't have anything to do with pharmacovigilance. It got into pharmacovigilance in a big way in 1987-88, and if you're familiar with this area, you probably have seen a CIOMS 1 form. That refers to the first CIOMS conference, which I believe was in 1988, headquarters in Geneva. ICH actually started as a tripartite agreement among the US, Japan, and EU, and its purpose was not really to assess risk. It was to harmonize regulations to improve the approval process. However, starting especially in the 1990s, both of those organizations got heavily involved in assessing risk as it became more important for medical products. The CIOMS consensus conferences are really thought leaders from agencies. FDA is always there. The other major agencies are always there, typically 3 or 4 large industrial companies, manufacturers as well, and then various experts from academia. So they're the thought leaders. They're telling us this is really what we ought to be focusing on. There have probably been about 10 CIOMS conferences that pertain to risk in this area. They're currently working on risk management. ICH then currently will take the CIOMS recommendations as published and will turn them into guidances. So they operationalize these ideas. Not all of these ideas can be operationalized. So ICH, which is essentially run by industry, but again, FDA is a signatory, promulgates final suggested guidances, which may or may not be enacted into formal law. They may or may not be published in the Federal Register. But from my viewpoint, it really doesn't make any difference, because since FDA is a signatory, FDA recognizes these international documents. They can be used for auditing. And if they are used for auditing, they can be used for warning letters. They can be used for citations, for 483s. They can become the basis of a CAPA. So from my viewpoint, it doesn't matter whether they're in the CFR or not. Next slide. So going back to some trends that are particularly important recently, I made a reference to case-based signaling processes. Hold on just a moment here. to case-based signaling using designated medical events. And what we mean by designated medical events is an important event which has a very low incidence. So an example might be serious skin conditions or hemolytic anemia. What's really interesting about the designated medical events is they show up over and over again with multiple cases, with multiple signaling situations with drugs and biologics. So if you will, they are drug-induced disease. This is what we're really talking about. Problem with them is they've never really been super well defined. Everybody knows what the core set of maybe 50 designated events is, but thereafter it becomes controversial. FDA Sentinel system, I think you may have heard of or aware of, and again, an important system. The problem for us in industry is, although it's a very nice system that allows epidemiologists to, at FDA to work up case-based signals, it doesn't provide information to us specifically unless there's a conduit through the regulatory pathway. The third bullet, I'll give you an example of a little later. Adverse event reporting systems and product complaint reporting systems, or quality systems, developed historically independent of each other. And there is a risk gap here, because information that really is could be an adverse event signal arising on the product side frequently doesn't make it very effectively to the adverse event side. So I just point out that is a risk gap, and that will become more important as we go forward as a result of biologics. Biologics are hard to manufacture. They're big, complex molecules. We're not talking about the simple molecules, pyrimidine rings, and so forth anymore. So I think one of the big risk gaps to focus on preventively is this gap between adverse event and product complaint data. And then, of course, increasingly we have signaling from risk management programs as well, really focused on well-defined events which are in that program. Next slide. So to tie all this up with some comments, first of all, CIOMS and ICH is where the action is in signaling. Safety signaling is not a one-size-fits-all. You can have a very large program with a product, and it can be non-compliant or ineffective. And you can have a very small safety signaling program with another product, and it can be quite adequate and in complete compliance. Signals come at you from all different sources. That, in some respects, is a risk gap, just trying to get all that information in the right place. Case definitions are really one of several concepts which are the heart and soul of safety signaling. And if there's one thing I would point to that I think translates into a problem over on the legal side more than any other, it's probably an inadequate case definition early on. Many companies don't do this very well. Looking for confounding factors, you'd be surprised the number of very, very subtle confounding issues which can appear to cause a relationship when that relationship doesn't really exist. Looking at the way the information system, particularly as it picks up things like publicity, can affect a signal and make it look much different from what it really is. And then lastly, epidemiological study results that April will discuss. Although studies are extremely helpful, there are many, many of them published around the world every year, and they're not necessarily well designed or well defined. There's not necessarily a good rationale for them. So don't overemphasize that word study. Next slide. So if I'm asked to evaluate a client, let's say from a preventive standpoint, what am I going to look for? First of all, signaling is a process. So my first inclination is to take a look at all the relevant processes and make sure that they're in place. If in fact there is something wrong here that ends up on the legal side, there are various considerations, but one would think if all the processes are in place, the manufacturer has carried out at least that side of their responsibilities quite well. So are they clearly defined? Are they being carried out by the right people? And if there's any credible information from either internal or external that warrants some sort of change to those procedures and processes, has that been done? When did the signal first occur? In the several legal cases that I've recently looked at, I actually don't think that the signal occurred when the plaintiff's attorney said it occurred, because I don't think that the systems that the company had in place, which were all compliant, would have had any opportunity to find that signal when it was proposed that they should have. Is the training and education adequate? Is a pharmacovigilance plan in place for pharma products that is a requirement worldwide? And are the systems appropriate to support the monitoring plan? Next slide. Now, I'm not going to go through this. This is just a quick data source checklist. But the point here is, if a signal comes to me, and I've done many of these down through the years in different settings, then sometimes it's very easy to say just at the case series level, not a problem. But once you get into a zone, a certain zone of uncertainty, then you really need to have other sources of information. And as you can see, there are many sources of information, potentially. So one key to working up a signal is to make sure you've covered these bases. Next slide. Now, if I was asked the rhetorical question, what should the jury know about this particular signal? I would go back to that question about processes. Did the client company have all the relevant processes in place? Did they have the right people in place? Were they identifying and disseminating information correctly? Did they respond quickly? And if all those things are in place, there really isn't much else you can ask it to do. They are compliant. You also need to have a clear, supportable case definition. Again, I think that's very key, and it should be medically oriented. You should know what the confounding factors are. You should know what kind of impact on the information system itself this information may have had, particularly publicity. I'll give you an example of that, and various factors that can affect study results. Next slide. So let's take a quick look at an example, Vioxx. This is a product that was pulled from the market, famous issue, legally and otherwise. And the real problem, as one goes back and looks at that signal, is in the second bullet. It was what some people uncharitably would have referred to as misinformation. Did the sales force, the publication strategy, just basically provide information that was wrong to the public? For the people, the written material that I've looked at that suggests that, if that's true, then the company painted themselves into a corner from the beginning, and I happen to think that that was a big problem with this signal. As we follow that through, though, and you look at these vascular event cases, heart attacks, strokes, every single one of them that was litigated that I looked at had multiple confounding factors. So it was a very weak signal from a case standpoint, if any signal at all. The epidemiologic studies that have subsequently been done suggest that there is an association. It's not a major association, and in fact, ibuprofen, well-known over-the-counter product, also has an association, which is more or less on the same level. So it also has a weak association with vascular events. There was an advisory committee held in the U.S. earlier this year, and they reviewed all that data and didn't say anything about removing ibuprofen. And plus, advisory committees in other countries have said, hey, let's put this product back on the market. So the bottom line to this signal, and unfortunately I think this happens more often than we might imagine, is make sure you have all the data, that you look at it from a signaling viewpoint, and don't panic and do something like pull a drug off the market prematurely, because you're basically telegraphing to everyone that you agree with the statements that are being made. Next slide. Metal-on-metal hip replacement is something I have been personally involved in, and I think the big risk gap here, and by the way, this is not unique to metal-on-metal hip implants. This is true for a lot of devices, is that the monitoring systems are very rudimentary for many different kinds of devices. That's a problem. That's a risk gap. Many of the so-called cases, if I used my definition medically, didn't qualify as cases. So some of them had infections, for example, that were related to the procedure. The study design for the EPI work was done outside the United States. You couldn't get that data, therefore you couldn't really replicate what they did. And much of the increase in reporting, and we were able to show this to the manufacturer we were working with, was clearly publicity-induced. In fact, the FDA later came out with a statement and said, this is a publicity-induced signal. There was no really good biological mechanism, not saying that what the litigators were saying was untrue. It may be true, but the biologic mechanism for it is iffy. So what lesson can we draw here? First of all, when you go to monitor a product, monitor them really excessively, monitor them at a level above what you're required to. Monitor them to the extent that you need to in order to reduce the risk for the product. And then secondly, I would say case definition is key. Make sure that that is put into place early. Next slide. GLP-1 anti-diabetic drugs and adenocarcinoma of the pancreas. Well, when you take a look at the case reporting, there's certainly the cases are there. David and I took a look at the FAERS database, and company asked us to do that. We looked at it and we said, yeah, there's some cases. It's an excess using the data dredging kind of algorithms that one would go to. But we also told the company, we're not really clear without a lot more information people like April can provide to us that there's really anything here. Why? Because there is a reporting bias, and it's not one of the usual ones. Anti-diabetic drugs act on the pancreas. Adenocarcinoma of the pancreas is located in the pancreas. And it's very easy for people to develop what it was defined back in the 1950s by the National Cancer Institute as an excessive sensitivity to situations like that, which is called ascertainment bias. Well, subsequent studies really haven't established that link with certainty. There supposedly are some studies that have established it. Other information has not established it. So you're really in a gray zone with this once again, which is exactly what David and I conveyed to the company would likely happen. Why? Because we saw an intrinsic bias in the reporting. I would also point out, by the way, that the incidence and prevalence of carcinoma of the pancreas is increasing. Most of the other tumors are decreasing, and that increasing incidence over time can also affect a potential relationship. Next slide. And then the last one I'm going to go to is multiple recalls of over-the-counter products that were initiated by an over-the-counter product manufacturer as a result of musty or moldy smell. And in this case, the allegation was that this musty or moldy smell was so bad and somehow had some biological effect that it was creating gastrointestinal events. People were getting sick. We had potentially an epidemic of gastrointestinal events on our hands. So the company initiated a recall, and then they posted, and I might say prior to this epidemic, they posted the suspect gastrointestinal events on their website. And what we found out from that primarily is when you tell people what to report, they report it. Because the vast majority of these events occurred in a remarkable up move on let's say a chronology line. The vast majority occurred after they were posted on the website. So the incidence and prevalence of background GI events are extremely high. I point that out because in this instance they easily could have been accounted for by background. Now, what we did in this case actually took advantage of the product complaint system versus the adverse event systems. Because they aren't well connected in most companies, that means we really have two independent sources of information. One is plotting out odor as a complaint, and the other one is plotting out GI events as an adverse event. We superimposed those plots, and anyone could have told from looking at that, as the FDA did, that there was no relationship between the two. So if, in fact, this odor is a harbinger of some terrible biological event, there's absolutely nothing in the reporting system that would support that. What did happen is there was a huge upsurge in GI event reporting after it was publicized. Now, just to hammer that point home, toxicity studies were subsequently done. And not only did the company, by the way, do a great job of isolating a contaminant that is environmental and it's been around forever and ever, and no one ever isolated it before, but they then did the appropriate tox studies, and it turns out it is completely biologically inert. It has no effect whatsoever on any mammalian system that they put it into. In fact, they could not reach a dose, no matter how much they gave the animals, that had any toxicity at all. So, of course, FDA was very impressed with all that, and the issue goes away. Now, what's the lesson here? The biggest lesson is make sure that you've got people who understand the ramifications of publicity before you begin posting potential events for other important related information. Next slide. And with that, I'm going to turn it over to April Zambelli.
So good morning or afternoon, everyone, as the case may be. I'm going to talk a little bit today about the use of epidemiological data in product liability litigation. Next slide. And I'm probably going to have to go quickly through some of these slides in the essence of time. So just to start out, types of studies and sources of data. By the time a drug or device is involved in litigation, there are likely many types of studies and sources of data surrounding the product. And the extent to that will obviously vary based on the specific product, the time in the marketplace, and so on. And we'll talk some more about some of these specific types of studies a little bit more in the future slides. But just generally, primary research involves the study or analysis of primary data, and what we mean by that is data collected or observed from firsthand experience. So randomized trials, observational studies, and registries are examples. And secondary research involves analysis of existing data, so usually, but not always, published literature. And then there are some studies that can utilize both types of data. Next slide. So analytic study designs are presented here. And what we mean when we say analytic study designs is that these types of studies are undertaken to test specific hypotheses about the efficacy and safety of a product. And an important distinction here is the difference between randomized controlled trials, which we call an experimental study, and observational epi studies. RCTs, or randomized controlled trials, are experimental because we can actually control who gets the exposure, or meaning the product, and who doesn't. Observational studies are different in that they're natural studies, and the researcher does not allocate treatment or exposure. We're actually observing exposure that's already taken place. Next slide. So just briefly to review, randomized clinical trials or controlled trials are conducted in 4 phases, and the term phase is really used to describe the goals of the trial. And generally speaking, they increase in size and duration and in the amount of and types of information that's generated. Phase 3 trials are what we call pivotal trials, and these are generally very well-controlled, randomized, of adequate size, and whenever possible, double-blinded, and these will form the basis of the application for FDA approval. Next slide. So strengths and limitations of clinical trials, and I'm going to just generally overview some of these for each of the types of study designs. The key strength of clinical trials is in their design. They can be used to evaluate causation, but the role or importance that they will play in that evaluation really depends on the question being asked and the nature and the design of the clinical trial. Randomized clinical trials are generally considered the gold standard for obtaining evidence of a treatment effect, and that's because they're a large randomized experiment, and therefore we can sort of randomize confounders and other important variables between the 2 groups so that the only difference between the groups is the treatment itself. Some limitations, and just because clinical trials randomized clinical trials are considered the gold standard, that doesn't mean that they universally trump all other types of studies. In fact, some of the limitations of clinical trials are presented here. They are generally designed to ask very focused scientific questions, and they don't always reflect real-world use scenarios. Examples include diverse populations or the effect of long-term use. There are also situations in which it would be unethical to conduct clinical trials. An example might be to give placebo for a serious illness where there's an established treatment, perhaps an experimental vaccine when there are already established and effective vaccines. Next slide. Observational studies. These are used to examine a wider range of exposures, typically, than experimental studies. As we said, they're natural studies that get around aspects of experimental studies which are not always feasible. One example, very typical example, is that an experimental study could not examine the effect of smoking during pregnancy on birth outcomes. As example, we wouldn't randomize people to smoking or not smoking, but obviously observational studies can look at something like that. They also mitigate many issues which are not feasible, as I mentioned, and some examples may include subgroup analyses. So, for example, randomized trials can often have very homogeneous populations, patient populations, and also the ability to look at multiple exposures and risk factors and to look at interactions between those variables. And an example there might be the ability to look at concomitant medication use. Next slide. So some of the strengths and limitations of observational studies. Just again, a reminder that observational studies draw inferences about the effect of a product on subjects when the assignment to the exposed or unexposed is outside the control of the investigator. And the most familiar types of observational epi studies are cohort and case control studies. Some of the strengths: real-world information on relationship between exposure and outcome, large sample sizes and long follow-up. As example, cohort studies can sometimes span decades. Different designs based on the question. For example, case control studies are often more efficient for studying rare outcomes. And they, as I mentioned previously, can often consider a wider range of exposures and confounding or modifying variables because they make efficient use of available data. Now, some limitations. Errors are inevitable in almost any epi study, even the most well-designed studies. And many of the errors or biases can be minimized by good study design or in the analytic approach. The key here is to be aware of potential errors and biases and to consider the impact of these on study findings. And examples of some of the challenges or limitations in observational studies include selection of the control group, standardization of measures, and missing data or data of low quality. Next slide. Post-market registries are a subset of observational studies, and unlike clinical trials, they allow assessment of product performance in a real-world setting. They contain data on large numbers of patients receiving care in diverse clinical settings and include clinical outcomes over time. Thus, they provide a really critical platform for capturing the experience with a product throughout both the product and the patient life cycle. Sometimes FDA will require a post-market registry, or a sponsor may voluntarily create a registry. And there are many examples of both product-oriented and disease-oriented registries. Next slide. And I'm not going to spend too much time on this slide, but it's here for you as a resource. Some of the strengths are similar to other observational studies. In addition, you can examine issues such as the impact of clinical experience or surgical skill on outcomes, so there is some additional information that can be obtained from registries. And there are limitations. Data are not 100% verified. You can have variability in data definitions, underreporting, difficulty in following up patients over time, incompleteness of data, and sometimes competing registries. Next slide. So the use of epi data in litigation. There are two types of data that are key to a causal assessment, and generally there's published data and then there's proprietary studies. And so obviously published data is available to everyone, but the degree to which the published data are representative of all the data available on a particular product will vary significantly on a case-by-case basis. And so not all clinical trial data will be published, and registry data, if initiated by the sponsor, will generally be proprietary. There will likely be observational epi studies looking at the safety and efficacy of the drug, some of which may or may not be commissioned by the sponsor. And internal analyses and studies may also be produced in the litigation. So given all the different sources of data that will become available during a litigation, what are some of the important things to do? And one of the important things is to really know your numbers. It's critical to understand from a very early stage what studies have been done and what data exists in what form. And then perhaps more importantly than the strengths and limitations of the data as it pertains to the question at hand. Accurate analysis of study data can address very important questions about potential safety signals, and I have some of those listed here. Pooled analyses and subgroup analyses are often very important in answering some of these questions. And so again, you need to understand the limit of the clinical trial data and to what extent the observational epi studies address these gaps or limits. Next slide. This is an example from a litigation in which I was a testifying expert for the plaintiff. This is excerpts from 2 data tables from the case, and just I'm going to real quickly go through this. And the numbers aren't necessarily important, but the issues that it highlights the importance of knowing the numbers. This was a retrospective pooled analysis of clinical trial data, and we were given a safety database and told that that safety database was complete. And I won't get into the issues, but it was not complete. And so in deposition, I was confronted with 3 additional studies that should have been included in the analysis based on inclusion and exclusion criteria. When that was brought to my attention, I assumed that the defense had already done that analysis and already knew the answer. But in fact, they did not. And when we went back and did the analysis, it only strengthened the signal in this particular case. And I was actually quite surprised by that. Next slide. Some other methodologic issues that will warrant consideration when you're reviewing the available epidemiology. Outcome reporting, lumping versus splitting, and I refer you to this FDA guidance document here that talks about the importance of how you classify outcomes or adverse events in studies, because whether you group them together or try to split them apart, that can really obscure or heighten safety signals. So that's an important consideration. Clinical protocols, monitoring and detection. So again, did the clinical trials have a priori protocols for detecting certain adverse events? If not, what are the implications of that? Does the epi literature address that? Inclusion and exclusion criteria, so how generalizable are the study populations to the general user population? And then length of follow-up, were the trials adequate to detect the adverse events or serious adverse events that are at issue? Next slide. So other important analyses that you should be doing with the data, sensitivity analyses. Is risk specific to a particular subgroup? Are findings robust to choice of control group? These types of methodologic decisions can influence the results and the inference that we draw from the epi literature. So it's very important to understand the numbers as they break out to particular subgroups. Analysis of registry data can be very useful for understanding the consequences of long-term use, as can epi studies if they're designed, to examine that and detect that. We can also ask if we see adverse events in previously unstudied populations or particularly vulnerable populations. And then, as I said, a review or analysis of the observational studies. And do we see replication? What is the evidence for pooled analyses or meta-analyses? Next slide. This is an example of a very high-level subgroup analysis, just to give an example. It's really just a graphical depiction to look for trends, so it's descriptive in nature. But in this case, this is a plot of the published clinical trials for a drug. It's an initial look at whether gender whether risk and benefit may vary by gender. So as you can see, based on, we have risk at the top and benefit at the bottom. As the percent of female study subjects increased, the risk for this particular adverse event increased and the benefit decreased. So we get closer to 0. So again, this is not causative, this is not definitive, this is just very descriptive in nature, but is an initial look that suggests that perhaps there might be differences in risk-benefit by an important subgroup, so in this case by gender. Next slide. And so this is a segue to the next section, and I know we're running out of time, so I'm going to go very quickly, but again, incomplete data can be very misleading, and so it's particularly important to consider what data have been published, what data have not been published. So this becomes a very important issue in published data. And that brings me to the next section. So information about publications will become available during the litigation, and that includes communications among authors, drafts of articles, and reviewer comments and feedback, among other things. And so it's important to ask questions about the publication record, such as what was the level of sponsor or corporate involvement? Did all the investigators have full access to study data? What findings were published, which were not published, why, and what can be discerned from the published data? Next slide. This is just excerpt from the Cochrane collaboration. For those of you who don't know, it's a global nonprofit really dedicated to synthesizing evidence with transparency and high-quality methods. And they did an analysis of sponsorship and found, in this case, that manufacturer sponsorship was associated with more favorable results. And this is just something that drug and device companies are confronted with, this notion of sponsorship bias. Next slide. This is a current example from the published EPI literature. In this case, it's testosterone therapy. And these authors did an analysis looking at the risk of cardiovascular events, separating the studies by funding source. And in this case, they showed a highly elevated odds ratio for non-industry funded studies, but not for industry-sponsored studies. So again, just another example of the types of analyses that are taking place related to funding. Next slide. And so I just point you to a resource by the International Society for Medical Publication Professionals on good publication practice for communicating company-sponsored medical research. And this was, these are recommendations from a steering committee. Next slide. I just provide some of those here. So, make the data public regardless of the outcome. So endeavor to publish the results from all the trials of marketed products. Make sure that all authors have access to all the data, study data, before writing the article. Make sure there's well-documented protocols, statistical plans and reports, data tables, and so on. So just some excerpts of their recommendations, things to keep in mind. And in conclusion, just to summarize, know the sources of data for your product, know the strengths and weaknesses of that data, understand the implications of the methodologic choices in generating and analyzing that data, look for data gaps, and review the publication record. Thank you very much.
April, thanks so much. And I just wanted to thank April and John for helping me put on this this webinar. Matt, I know we have a hard stop at 2:50, and I guess we don't have time for questions, but I guess I encourage everybody. We'll be sending out the slides, I guess, in an email tomorrow. Is that right, Matt?
Absolutely. I want to thank you so much. Thank you so much to all of the presenters today. We will be sending out the slides tomorrow, as well as a link to the recording. We probably can fit in one question. We did have one question come in. Dr. Clark, you mentioned the importance of case definition. Can you expand on why that's such an important factor?
Yeah, this is John Clark. I think that came out of my presentation. The purpose of the case definition, it's actually multiple reasons, but one of the main ones is so that we can do subsequent analyses. Now, if you end up with multiple parties counting different things, which I have seen, particularly by the time it gets to a legal environment, it's entirely possible for one party to end up showing something which is at odds, even opposite, of what another party shows. So laying the groundwork and challenging people and saying correct definition. Now, if you don't want to use this, you better have a pretty good reason for that. I think that approach is essential in this entire process.
Okay, that's fantastic. Well, with that, I want to thank you all for your presentation. I want to thank the attendees for your attention this afternoon. And if the panelists want to say a quick goodbye.
Thank you very much. We appreciate your attendance.
All right,
thank
fantastic.
you.
Well, thank you very much again, and you can expect an email from me within the next 24 hours or so with the link to the presentation as well as the slides. That concludes this presentation. Thank you very much indeed. Have a great day.